Hi all and welcome back to this second part of Beyond Multivariate Analysis, an introduction to the formal encoding models. In the previous section, we were understanding how brain encodes information, and we noticed that information can be encoded in the form of patterns, but also in the form of activity profiles. We understood that depending on the technique that we use, we can be closer to the information representations, or we can be closer to neural activity. Now, in the second part of the course, what we will do is to understand how the encoding models work, and we will see different examples of how they can be used. To understand what is an encoding model, first we will remember what a decoding model is. As you probably know, after doing this course, you have learned that to test a classifier, which is kind of decoding model, what you have to do is to present a participant with different stimuli, then you record activity from a part of the brain that is interesting for you, and then you train one of the classifiers to maximize the difference between the different classes that you want to classify later on. Once that you have trained your classifier, the next step is to cross-validate to avoid overfitting, and then what you do is to take data from the brain in another registration session, and what you have to do is to verify whether using that brain activity you can classify the different stimulus correctly. If you are able to classify the stimulus correctly, that means that there is information in the neural activity that can be used to differentiate between different classes. So essentially, a classifier and decoding implies to go from neural activity to the stimulus. This is what is called a discriminative model. So it's discriminative because what it basically does is to separate between the different classes. It tries to maximize the difference and say if something is different or if something is similar, without really understanding why the two classes are different or similar. In contrast, the encoding models have a different purpose. Although some parts of the functioning are similar, there are some critical points that should be taken into account. Here we will oversee a little bit what is an encoding model. So basically what you do with an encoding model is to first record the brain activity when participants are seeing a different stimuli. In this case, I'm using gratings, but you can use naturalistic stimuli. So what we want to do here is try to explain and understand how each one of these stimuli are producing the activity that we are recording here. One of the advantages of a linear space, or assuming a linear space, is that we can use a linear regression. So if we imagine that brain activity is Y and the trials, which are the regressor in a multiple linear regression problem, as you can calculate the weights just using the brain activity measure and then using your regressors, which are the stimulus. And then once you can get the weights, the last element that you need is the noise, that you can also calculate the noise based on the weights, the Y activity, and the stimulus that you have presented. So what is the next step once that you have to train your model to predict brain activity? You have to test whether your model is actually generalizing to new stimuli. So what will be one possible test? Similar to cross-validation, what you do is to present new stimuli to the participant. In this case, imagine that you are presenting a Mickey Mouse cartoon, and then you can decompose this Mickey Mouse cartoon in smaller feature, because as you can imagine, each image can be decomposed in a sum of different smaller features like gratings, and then you can based on the model that you calculated previously, you can estimate what will be the neural activity evoked by this stimulus. So now that you can predict neural activity, what you have to do is to record the actual neural activity evoked by the stimulus. And if both the predicted and the measured neural activity are similar, that means that your model is working good. So the difference between the coding models and the encoding models is that in the encoding model, what you do is to learn how to map the stimulus to neural activity. These type of models are known as generative models. Some of you might think, okay, this is a little bit boring because I'm interested in knowing or in the coding activity from the brain. So I want to know if I can separate between different stimuli. Okay, don't worry, because using the neural encoding models, you can invert your model, because once that you have learned how to map from stimulus to neural activity, you can invert your model and use it, for example, like a decoding model. So this is the example that I showed you before, where we have like trained our model and we are able now to use this model to predict what will be the activity evoked by this Mickey Mouse based on how we train our model before. So what we can also do is like use our model to predict what are the features that compose this that evoke this neural activity pattern. And finally, as we understand what specific features have been evoked by the neural activity, we can not only classify the stimulus, but we can also reconstruct the whole visual stimulus. So probably using this forward encoding model, we will be able to recover the image that the participant is seeing, like an entity and not like just a category. To illustrate this, I think that it would be better if I leave you with Jack Gallant, which is one of the leading scientists in using this technique and one of the pioneers that really take advantage of encoding models. So here you can see the video. It's also on YouTube.
My name is Jack Gallant. I'm a professor of psychology and neuroscience here at UC Berkeley, and my lab works on vision. That's our main focus, is on the visual system. And vision's a really important thing to work on because humans are very visual animals. And when humans have damage to their visual system, they're really incapacitated. And natural vision is a little like watching a movie. So essentially, we were trying to build a model that describes how your brain responds to movies. So if the external world is a certain kind of movie, we want to be able to build a dictionary that will allow us to accurately predict what the brain activity would be that would be evoked by that movie. And then given some brain activity, we want to be able to reconstruct the movie that you saw. So on the left here, we have a movie that we showed a subject in the magnet. And on the right, we have a brain. And you can see the brain activity is painted here on the side of the brain, but the brain's kind of crunched up in the skull, so it's hard to visualize. So we kind of inflate it, and then we flatten it out, and we can see a flat map of the visual cortex and the rest of the brain as the person watches the movie. The problem here for us is to translate between these movies that occur and this pattern of brain activity that occurs. And in the study we talked about in our paper, we just modeled this very back part of the brain, the early visual system, primary visual cortex. And that part of the brain responds to the little local features in the movie, little edges and colors and pieces of motion and texture. But this part of the brain doesn't know anything about what the objects are in the movie. First, we put people in the magnet for several hours, and we show them movies, and we build a model of their brain. And then in the second part, we put them back in the magnet, we show them new movies, and we measure their brain activity, and we decode their brain activity in order to reconstruct the movie. So on the left here is the movie we actually showed people, and on the right is our reconstruction. When the movie that we showed has a fairly common object like a person, our reconstructions are actually fairly accurate. But when the movie that we showed is something rarer, as you'll see in a second, like this abstract thing, then our reconstructions are coarser. Not really so much over here, I guess. I think you'll be able to use this kind of technology for communicating with people who can't communicate, like locked-in patients or people with neurological diseases. I think you could maybe use this in some forms of psychotherapy or dream analysis. I think there's a lot of potential clinical applications for this. When this model worked, we were very happy because it was really quite surprising that it would work at all. We're only taking snapshots of the brain every 1 or 2 seconds, and our reconstructions of movies happen at the movie rate. So that's much faster, and there's no reason one would think that this would work. And in fact, most of our colleagues just told us this would never work. So it was pretty surprising and satisfying when it actually worked.
So as you can see, it's very impressive how useful this technique can be to decode information from the brain. So now let's dig in a little bit on how it works. So let's see with a little bit more of detail what are the different steps that can be applied in order to use this technique. How forward encoding model works, we can divide this in two main steps, very simplified version of the functioning of the model. The first step will consist of linearizing our external world. We are assuming that the neural space is linear. This is something that is not realistic. We know that the world is not linear, but we need to make this assumption in order to simplify our calculation. On the other hand, and first of all, what we do is to transduce the sensory information to a linear space. In this case, you can see this Homer Simpson cartoon, and we need to project it onto a linear space composed by different gratings. This process is called linearization. Okay, so in an abstract way, we can see this figure here, and it will mean that you can represent each one of these pixels that conforms the figure as different weights that component different predefined channels, in this case orientation. So let's, for example, imagine that we take this pixel here. So probably if we project this pixel information across our predefined channels of orientation, what we will see is that this channel here has a strong weight because it's really defined by a diagonal clockwise oriented information. Then this one will be almost null weight. This one has a little bit of weight, and this one will be a little bit of weight too. So we can have like a profile of a feature weighting that can be represented here. So once that we have done this for all the pixels of our image, so in the end we have like a full representation linearized of the image that we want to decode, what we have to do is to proceed with the linear mapping. So the linear mapping is basically see how much of these channels that we have recorded in a trial by trial basis are explaining the neural activity that we have measured from the participant brain. To do that, what we use, as I said before, is a linear regression model. So we can do this for each voxel or each neuron, or we can do it even for each set of orientation and use all the information here as a multivariate pattern in order to have a relationship between orientations and neural activity. Once that we have learned what is the forward solution of our model, we can start to decode or to see if the activity that we are measuring is really matching the activity that is predicted by the model. Okay, now we will see one example of how this technique can be used, and this is one of the pioneer works using forward encoding models with naturalistic images by Kendrick Kay and also the Jack Gallant lab. So what they did was to present the participants with a lot of naturalistic images. As you can see, the sample size of participants is only 2, but what they do is to train these participants and test during many days for long sessions, so they can like really build very accurate and precise models of brain functioning and their correspondence to the stimulus space. And they present this in a scanner of functional magnetic resonance, and it's basically as simple as this. They are like looking at the screen, and they are like images projected for 1 second on and off 200 milliseconds. and then some resting space. So the first problem that you have to account when you want to linearize the space is to choose how you are going to linearize the space. This is important as you cannot do it trivially, and this is one of the important potentials of forward encoding models, and is that you can model brain activity using neurophysiological inspired models of how the brain should work. And we know that in visual cortex we have some neurons that have different receptive field sizes, and they are like tuned to different positions of the visual field. They are tuned to different orientations of the visual field and different spatial frequencies. So to mimic this behavior of the neurons in the visual cortex, what they did was to pass all the images through a set of filters that they mimic different receptive field sizes in different locations on the visual field and with different orientation. And this is what is named as a Gabor pyramid wavelet. And what they do is to calculate what is the combination and what is the weight of each one of these components that better represent the image that is here. So they do this for all the images that they have, and this way they can linearize the space. So it's represented in visual receptive field maps. So once that we have the weight for each one of these stimulus features or channels, if you want to call it like that, which is carried out by a non-linear minimization procedure. So basically what is trying different combination of weights until the combination of weights is giving the largest amount of fit to the image presented. The next step is to see how the neural activity in the brain is explained by each one of these channels that we have pre-created under weights. So then we have for each trial a set of stimulus channels, and we want to see whether at the trial basis this activity is explained by the combination of features that we have created. Now, for example, we can focus on the activity profile. So if we are looking at the voxel level or at the neuron level or even the electrode level, or we can do it at the activity pattern. So then you train your linear model and you can recover the weights and the noise. And what they did there afterwards was to, here a very silly example, but they present an image, then they look at the activity that is produced by the brain when they see this image, and then using the model that they pre-train, they try to predict what is the activity for each stimulus. And this is another image, and this is activity for all the voxels that is supposed to be produced by the brain based on the model. So this is predicted activity, this is predicted activity for another stimulus, and from the same stimulus that we are looking at the activity here. And as you can see here, this activity and this activity is very similar. So that means that it's working pretty well. So it's predicting the activity pretty well, and also you can use it to classify, because if you have to select what of these stimulus was presented, what you have to do is to say what of these neural activity profiles is more similar to the activity profile produced by the image. In terms of performance, what you can see is that this kind of modeling technique is giving you a very good performance. So this is the correlation between the pattern activity evoked by the predicted image in the y-axis and the pattern of activity evoked by the actual image. And in the diagonal is the unity line, and as you can see, the correlations are pretty close to 1. So that means that is giving a pretty decent neural patterns that are like super similar to what is predicted by the model. And finally, we can like check how is the coding performance, so how good is like in separating between the different stimuli. And what you can see is that if you are using repeated trials, so that means that you are like training and decoding on not only one presentation of the stimulus, but many presentations that you average out, then you can like do it about chance level and see that if they use, I think, 120 images, chance level will be this small portion here, right? The green bar in the bottom part of the plot. So the average of the coding is y above the chance level. And what is very interesting is that only with one single trial presentation, the decoder can classify also very much above chance level the images. So if you are like training your model and you just give to the model one stimulus presentation, it can very accurately say or tell what is the image that was presented. So that means that this speaks very well about the performance of these forward encoding models. Okay, some interesting properties of the forward encoding models are, for example, that you can decode activity at the neural or voxel level. For example, let's review some properties that could be tested with this type of models, and indeed Kendrick Cake did in their study. So what we know about the visual cortex is that the size of the receptive fields are increasing the farther away we get from the fovea. That means that when you are close to the fovea, the neurons have like a smaller receptive fields, so they have more precision to the small details of the image. And when the image is more in the periphery of the fovea, then our capacity to see the image is lower and the receptive fields are larger. This is a property that using, for example, retinotopic mapping can be recovered. So this is one of the techniques that also can be used with forward encoding models. But here we can see that orientation is represented in visual cortex and also the eccentricity. And the fovea has a lot of neurons, which is the yellow and red part, dedicated to the processing of the image, and the periphery has less neurons. This is called also cortical magnification. This characteristic that larger visual fields are presented at the periphery of the visual cortex can be recovered in Kendrick paper. What they observe is that for those neurons that were more tuned to the periphery of the visual field, they also had larger receptive field sizes in V1, V2, and V3. So that means that this is a property that we know from a visual system and from the neurons in the visual system, and using this with forward encoding models and fMRI can be recovered. Also, the orientation tuning that we were expecting can be recovered using the forward encoding model. And what they found is that, for example, you can take one voxel in visual cortex, and you can see that this voxel really likes, so this is the image, right? This circle is the visual field of the image. And then this is the tuning, the spatial tuning of the voxel. So the voxel likes things that are presented in this part of the visual field. And also, what you can recover is what is the orientation tuning. And in this case, this neuron likes more 45 degrees orientation stimuli, and also is tuned to relatively high spatial frequencies. Okay, so we can recover both the receptive field size, we can recover, which will be given by this tuning, we can recover the selective spatial tuning, and we can recover the orientation and the spatial frequency of each voxel. Another cool property of the forward encoding models is that you can model as many dimensions as you want in your data. This is another example of applying forward encoding models to other type of stimuli, in this case language. And imagine that you have the participants listening to the sentence. How do we define it? So what you have in the beginning is an audio waveform. right? And this audio waveform can be decomposed in an spectrogram, which will give you the amplitude for each frequency band and in time. So these are some features that you can introduce to your model, that you can even use a Gabor filter as we use with the images to model the phonetic or the acoustic characteristics of the presented stimulus. But you don't need to limit yourself to use only the acoustic characteristics of the stimulus. You can maybe use another feature that is the envelope, or maybe you can go higher and use the phoneme deconstruction, and you can go even higher and use a word deconstruction. So all these features are supposed to produce activity in the brain, so they can easily be introduced in your model, and then you can investigate how much each part of the brain is like responding to phoneme, envelopes, words, or the acoustic characteristics of the stimulus. Adding new dimensions to the model can help to explain additional variance. For example, if you have this target image and you want to classify it, your classifier might commit an error and say that this image is this one or this one. So it depends on how you train your classifier, will commit different types of errors. For example, if you are using just a model like the one that Kendrick K used, and is based only in Gabors and the structural distribution of the image, many times your classifier will tell you that this image is this one. But for example, if you add a semantic model of representation of the image, then the type of error will be different, and possibly instead of like saying that this image is this one, will take this other one that it contains some building representations inside. Depending on what information features are you introducing in your model, you will make better predictions. So in this case, if what you want to do is to classify objects as animate or inanimate, in this case possibly is going to fail more often, because it's only taking the structural information, and probably to classify inanimate from animate is more important in semantic component. In fact, what they saw is that comparing a model that only explain information based on wavelets and another that is predicting information based on semantics, the two models are explaining very well the information that is presented in the images, but the semantic model is explaining information that the Gabor wavelet model is not explaining, and vice versa. So the combination of the two models is going to give you in the end the best performance. Another interesting property of the forward encoding models is that it allows to compute what is the upper limit of the coding in your data. Logically, the prediction of the model is never going to be perfect, as your data have noise. For example, there are also fluctuations in the tension or the activity of the scanner. Anyways, you can try different types of model, and you can, like, in the end see what will be the upper limit and compare with different versions of the model whether your data predictions are improving or not. To illustrate that, I will use this study by Hatt and Galland lab that was run in 2016 and has some impact in the media also, where they recorded data from 7 subjects, and these subjects were listening to naturally spoken stories, and this was like in the previous experiment that I described, for many days of recordings in the scanner. And what they focused on the end was in the semantic characteristics of the speech. So they decompose the speech in semantic features, so they could like get some regression weights and then see how they explain the ball responses. And then they had a new story and use that in order to predict the information that is presented in the brain. So that way they can compare what is the performance of the classifier. So depending on the good correlation between what the model predicts and what the activity of the brain is, then you know how good your model is performing. So here I'm gonna show you a map of the accuracy of the model, and you can see that in different voxels the model is performing quite well. For example, in some parietal regions and temporal lobe and frontal lobe, while for example in somatosensory and motor regions the performance, as it would be expected, is not really high for the encoding of semantic contents. So interestingly, then we can like go and dig a little bit more deep and see what is the representation of semantic information in the brain. And what they found is that there are like different dimensions in the semantic space that have a lot of representation in the brain. These are, for example, the social components or the visual or somatosensory components. And these semantic maps are not very homogeneous, so they are actually heterogeneous and they are spread across the brain. So there is no region for social content, or there is no region for visual semantics, but you can see that it's quite spread along the cortex. But what is interesting is that they saw that in most of the participants, the regions show a prominent overlap. So the map is quite systematic across participants. If you are interested in exploring a little bit more by yourself the organization of these semantic maps, I recommend you to click on this link here, and for example you will see something like this, which is the atlas. It's an interactive version of the atlas generated in the paper. You can see the different regions, and we can spread and unfold the cortical layers like this. Here you can inspect with more detail, for example, this parietal region and what is representing. In this case, we can see that the model is pretty reliable and there is a strong significance of the model performance, and those concepts that are like encoded in this part of the brain are related with social aspects of life. The visual ones, this is super reliable, it seems. So here we have some textures which really overlap with tactile and visual. Now, to finish with all this part of the forward encoding models, I'm gonna tell you that forward encoding models is not something that's super new for most of you, and probably you have used them before. So one example is the GLM applied to model blood oxygen level dependent signal, the Bolt signal in the brain, and what basically you are doing when you model Bolt signal. So you have some fMRI data that is recorded in these GPI sequences, and then what you have are your features, which are your experimental manipulations in the experiment that you will be using as a regressors, and then what you want to calculate is the beta parameters. So in the end, what you want to do is to build a model that is able to predict Bolt signal modulations in the time series. So here in red we have the actual signal, so blue will be the model predictions. Another everyday example of the use of forward encoding models is source localization. For source localization, we apply dipole fitting or beamforming, which is some kind of forward encoding model. And as you know, in the inverse problem, what you want to do is to localize where are the sources that are causing the activity registered in these external electrodes, right? So to do that, we need to formulate and forward encoding model, where we want to model and predict what will be the activity in each of these layers, considering different weights for each one of these features, and taking into account also the structural anatomy of the brain and the skull. So when you solve this model, what you get in the end is some kind of a spatial filter, and you can localize where the activity is, and also you can even project back this activity from the external channels into these virtual channels that are like created inside of the grid of the head. Thus, as brief final summary of this theoretical part of the course, I would like to go a little bit over the different techniques that we have been using before, and that involve decoding and involve encoding. First, we can talk about pattern component analysis and multivariate classifiers. So what we can say is that they will give you a good sensitivity to classify manually labeled classes at the cost of poor understanding of what we are classifying. Then we will have a low spatial resolution. So the spatial resolution will depend on the number of boxes that you are, or electrodes that you are using on your search slide, for example. It doesn't aim to explain neural activity. So the only thing that it tries is just to separate between neural activity and see if the patterns are different. And it has the advantages that it's quite easy to implement. So you don't really need to think too much. You just feed the model or the algorithm with the different patterns. You give the labels, and then you cross-validate. And if it's over the chance level, that means that there is information. Then we have the representation of similarity. This case is like some kind of extension of the multivariate classifier. So as I mentioned before, remember that you can use correlation or you can use Mahalanobis distance, but if you want to train a multi-class classifier and you look at the confusion matrix, so which classes are more confused with others, then you can also create RSA, so you can like see what is the dissimilarity between the different classes. So in the end, it's more or less the same, but it's gonna give you a representation of the geometry, of the representational geometry of the stimulus that you are using. It will allow you to test different models of brain representation. So you can, this is really good because at least you can like have some hypothesis about how the information should be related in the representational space, and you can test if this happens and where does this happen. So this will be akin to linearize the stimulus space as we did in the forward encoding models. The RSA, as the classifiers do not aim to explain neural activity, only to see how different or similar the representation, and it also has the advantage of being easy to implement. And finally, what we have been learning here are the forward encoding models. So the decoding accuracy will depend on how good is your model. It's really useful compared to classifiers or even representation of similarity to decode naturalistic stimuli, because you can fit as many dimensions as present in naturalistic stimuli. See that naturalistic stimuli is super rich because they are rich in semantic and sensory content. It can generalize to new stimuli as well, as what you are doing is to decompose stimuli in simple features and then correlate the neural activity with the simple features, and then you can like see how these features recompose a new stimuli. Then the objective of these encoding models is to explain neural activity. As we said, the coding model goes from activity space to stimulus space, and neural encoding models goes from stimulus space to neural space. And then it comes with a difficulty that is more complicated to implement, because you have to think how you are going to linearize the naturalistic stimuli, and that sometimes is a challenge. Here I'll leave you with some relevant literature. I think that some good papers to read is this one from Christopher Holdraff, and it goes making a good review of encoding and decoding models, and also will give you the opportunity to try some Jupyter notebook examples. So it's not only the theory, but you also have some practice to do, applied to language. Then this other paper from Naselaris. Then this other paper from Naselaris is also very interesting and simple to understand, and is more to the conceptual contents of the forward encoding models and what are the advantages. And then in general, I recommend this tutorial written by Pim Moster, where he describes a little bit what the decoding models are, how they work, describing in very detail linear discriminant analysis, and also you can see something about generative models. This is the author also of the toolbox that we will be using for the practical session, so I think that you can take a look at what he has written. I think it's very clear, and he gives very good practical tips.